All right. Welcome everyone to this December Ask Me Anything. My name is Josh. I'm here with our Director of Patient Services, Shannon Lozinsky. Um, I see some familiar names and faces, so we can open it up to questions. So, Sarah, over to you.
Yeah, thank you everybody for being here. It's so good to see some of you again, some for the first time. And I'm very thankful to the people who sent in questions via email ahead of time. So I think we'll probably kick off with one or two of those. But just like Josh said, we really encourage you to raise your hand if you guys have a question here too. So let's start with one that we got via email, which is Shannon and Josh, what's one thing that each of you would want a newly diagnosed patient or their caregiver to know? What's like the most important thing you'd want them to know right away?
Well, Shannon speaks with people every day. So maybe Shannon, I'll turn it to you and can add anything else.
Sure. So I think what myself and Andrea and Kimberly, our patient navigators, the things that we always want to relay to patients are that finding the right medical team that has experience with chordoma in whatever way you can is very important, and that in most cases you have time um, to be able to look around to talk with different doctors and figure out what you'd like to do, and then third, connecting with others just to find the support. I think the thing we hear all the time is that it can be very, in addition to getting a diagnosis of cancer, you're getting a diagnosis of a rare cancer, which can feel very lonely and scary. And so the relief that we hear in people's voices when they connect with others who have been through what they're about to go through is huge, and that can really make such a big difference. And so those are kind of the standard things that we always encourage them to do, and then always remember that we're here also, and that you can always turn to us, and we're here to help in any way we can.
Yeah. Could not agree more with all of that. And I think just to build on that and relatedly, um, I think that this is a disease where kind of being thoughtful and methodical and taking your time really pays off. So there's a lot of ways to make a misstep, um, and the reality is that oftentimes patients get differing opinions, and it's not necessarily that one is right or wrong, but oftentimes what is right for you is based upon a lot of different factors, and maybe not every clinician has the full picture. So it's really important, not only to be to seek experienced clinicians and experienced care team, but to seek a multidisciplinary care team that where the different specialists are going to coordinate with one another, and then to get multiple opinions and to kind of compare, cross-reference, triangulate, if you will, and ultimately make a decision or decisions that are right for you based upon your particular situation and preferences.
Mhm. Thanks, you too. That's really helpful. And I just want to add, I'm about to paste in the chat a link to our patient navigation service. So that's a free service that we offer where you can speak one-on-one and get personalized guidance from our patient navigation team. And so if you're newly diagnosed or your loved one is and you're feeling overwhelmed about the options that are available to you, we just really encourage you to reach out to our team and get some more help. Okay, next question is a patient wrote in saying they'd caught wind of the new results from the pemetrexed clinical trial, and they're wondering, Josh or Shannon or both, if you could just speak a little bit to what that study was about, the significance of those results, and where we go from here.
Yeah, absolutely. It's very timely and very exciting. So for those who haven't seen, maybe we can actually paste the blog post into the chat. There's a small clinical trial that was done over the last several years that the foundation co-funded along with the pharmaceutical company Lilly, testing a drug called pemetrexed, which is a widely used chemotherapy drug that's been around for decades. It's used to treat a number of different tumor types. The A lot of oncologists have experience with it. The side effect profile is very well known. And there was some kind of hints that maybe about 4 or 5 years ago that this drug might have a role in chordoma. And a clinician in Southern California named Dr. Santosh Kesari, who, based upon kind of the hypothesis that he had, treated 3 patients with pemetrexed. These are all patients who had advanced chordoma. And 2 of those 3 patients experienced a notable benefit on that drug. And so based upon those results, Dr. Kesari came to the Chordoma Foundation, and we had a discussion with our medical advisory board about exploring this concept of pemetrexed in a larger number of chordoma patients. And so ultimately, we co-funded a trial testing pemetrexed in 14 chordoma patients. And as reported earlier this week, the results are really quite promising. In fact, with the caveat that this is a small study, the best results we've seen to date from any drug for chordoma patients with advanced disease. So specifically, of the 14 patients who were treated, 10 of them had their tumors shrink to some degree. 2 of those were considered significant. So there's kind of a generally accepted threshold in oncology that applies across many tumor types, that a reduction in tumor dimensions of 30% constitutes what's called a partial response. And there's historical reasons why that 30% threshold is used. There's a lot of debate about its appropriateness for all tumor types, including chordoma. And I think many oncologists would argue that it's maybe not the best measure of significance, but nonetheless, that's what's generally accepted. So you have 2 out of 14 patients that achieved that threshold, so roughly 14%. For historical comparison, other drugs that have been used to treat patients in the advanced disease setting, drugs like Gleevec, for example, which probably many people have heard of, have a response rate. So they achieve a RECIST partial response, so the 30% reduction in tumor size, somewhere less than 5% of the time. So this seems to be, again, with the caveat that it's a small trial, seems to be more active than drugs like Gleevec. But I think even more importantly, many patients on the study reported quality of life improvements, improvements in vision, reduction in pain, improvements in mobility, things of that nature. So I think it's really promising, promising in its own right, but additionally promising because this drug is used in a number of other tumor types in combination with other drugs. So it's like serves as a backbone for combination therapy. And so right now, in parallel, there are a set of experiments that are ongoing in Chordoma Foundation labs to try to understand whether this drug, pemetrexed, might be appropriate and could be synergistic with other drugs in combination. And then in addition to that, there's a clinical trial that's being planned right now testing pemetrexed in combination with an immunotherapy called pembrolizumab, so commonly called Keytruda. And some of you may be familiar, but there was a clinical trial done in France that was published, I believe it was earlier this year, Shannon, showing actually similar results with Keytruda. So Keytruda and pemetrexed now are kind of the drugs for which there is the greatest evidence of response in the literature. Those 2 drugs have been used in combination in lung cancer, and there has been synergy observed. And so Dr. Kesari has come back to us with a proposal, and our medical advisory board has approved that proposal to test that combination in an upcoming clinical trial. And we're just kind of stepping through the various administrative hoops to be able to get that trial off the ground. But I think there's great potential there. But even short of that, those combination trial results, I think this is definitely something for those who might be in need of a systemic therapy that would be worth talking to your oncologist about. It represents potentially a new tool in the armamentarium that we now have against chordoma. The final thing I'll say is that ultimately all of these drugs are going to need to be applied in a personalized way. So, a subset of patients seem to be benefiting, but we need to figure out which patients are benefiting and why, and then use that information to apply these drugs in a more targeted and personalized manner going forward. So that is an additional area of ongoing research. And ultimately, I think, as is the case for most oncology drugs, unfortunately, tumors evolve and they find ways to get around drugs that are thrown at them. And so there is still a great need to find additional drugs that could be used either sequentially with pemetrexed or in combination with pemetrexed to improve the response rate. Shannon, anything that you would add to that?
No, I think you covered all of it. Great, thank you.
Great, thanks. And you can, you can find more details on this stuff in the chat. And I also just wanted to say, some new people joined the call since Josh did the housekeeping notes at the beginning. So just to recap how to ask a question, you can use the raise hand tool, which is located at the bottom of your Zoom screen, or you can type your question into the Zoom chat and we will get to it there. Meanwhile, Richard, you have your hand raised. Go ahead.
Hi, Josh. Hi, everybody.
Hey, Richard, good to see you again.
All right. So I was reading, I think I joined this Facebook group with a Chordoma survivors, and there's this young girl, 16 years old, where the disease got into her lung area, and it was so sad. And you say, I hope she can make another Christmas and stuff like that. It kind of broke my heart. But the question got to ask you is, why do you think I don't know if it's that part of the body, but the chordoma is more susceptible to the lung area? And a follow-up question is, what is the consistency of the chordoma? Because I've heard it's like a jelly-like substance, but if you could maybe just address the first part, why is the lung area? Because when I had my sacral chordoma, I had to get a CT scan of my chest, and I continued getting them. So why do you think that's, it's prevalent in your lung area if it spreads?
It's a really good question. I think the short answer is that we don't know at the moment. I think an open area of research right now, or an important area of research, is understanding the metastatic process in chordoma. So most chordomas don't metastasize, at least early on in the natural history of the disease, or like early on in the disease course. But over time, a not insignificant number do metastasize. On the other hand, some patients actually do have early metastases, and even metastases at the time of diagnosis. And so understanding why that is pretty important. And we're supporting some research studies to try to compare metastatic tumors with non-metastatic tumors to understand, are there fundamental biological differences? But at the moment, we're just scratching the surface. So it's a good question, but it's one that further research is needed to answer. And then as far as the consistency, you're right. I think chordomas are oftentimes referred to as gelatinous, but from my understanding, I think this would be a question probably better directed to a surgeon, but from my understanding, there can be significant variation in the appearance and consistency of the tumor. Whether that has any particular bearing on kind of the clinical course of the disease, we don't know. But also it can change pretty substantially between a primary tumor and a recurrent or metastatic tumor. In particular, it's often the case that like a recurrent tumor might be kind of more kind of involved in scar tissue and things of that nature. So it might be the borders of the tumor and the kind of the tumor capsule might be less defined in case of a recurrent or metastatic tumor. But I think that I would probably defer that question to answer any further detail to a surgeon. Shannon, anything that additionally?
No, well, nothing additionally to answer the question, but we don't know, like Josh said, we don't know why it spreads to the areas that it typically spreads to, but we do know areas where it does typically spread. And so, Richard, like you said, you've been given chest scans, and you do that regularly, and that's because we know that the lungs are a very common place. If it's going to spread, that lungs is a place where it does go. We also know that lymph nodes and the liver and skin, and then other bones. And so some of that seems to be connected to where the original tumor is located. So if you have a tumor on your sacrum, maybe if you had spread to other bones, it could affect your pelvis. Or if you have a thoracic tumor, maybe it's more likely that it would spread to your lungs just because of the closeness of it, or, you know, things like that, that, like Josh said, we don't necessarily know for sure, but patterns kind of have been observed. And so when you have your scans, you may not have a chest scan every time you have an MRI of where your tumor site was. You may not also have a full spine MRI every time, but those things should be done periodically just to make sure that there's no changes there. So Richard, you know, that's kind of why you see, and they do look for that when you're first diagnosed. You should have that full spine MRI and a chest scan to make sure that there are no other, like Josh said, it's very rare for there to be metastatic sites on diagnosis, but it can happen. And so those are the things that you want to just keep an eye on and make sure that those scans are happening so that there's no surprises.
Great. Thank you, Richard. Really good question. Thank you so
Yeah.
much. Another question offline. Josh, this one's probably for you. Why did the Chordoma Foundation decide that it needed its own lab, and how have things been going there?
That's a great question. So, you know, ultimately, what we're after is better treatments for this disease, and there's a number of different ways that a number of different modalities that might represent opportunities for better treatments. But the primary one is systemic therapies, so drugs, basically. And the process of finding drugs that work for chordoma, it involves a whole series of steps. You could think of it from A to Z. So it involves, just to kind of briefly summarize, understanding the biology of the disease, letting that biology of the disease guide us to hypotheses or ideas about potential weaknesses or vulnerabilities in the tumor. That either might point to a existing drug or drugs that could be applied to chordoma, or it might necessitate developing new drugs. And then ultimately, there needs to be clinical trials testing those repurposed or those new drugs. So there's a whole set of things that need to be done. Now, there's certain of those things that are really well suited for academics. So discovering fundamental aspects of chordoma biology, identifying new therapeutic targets. There's researchers who are kind of world leaders who have niche expertise. You know, they're the leading thinkers, not necessarily in chordoma, but, you know, have relevant capabilities, expertise, approaches, et cetera, that we want to bring to bear on this disease. At the other end of the spectrum, you have medical oncologists who are going to be involved in running a clinical trial, and you're going to be, you have companies that are going to need to be involved in providing drug and hopefully funding for those clinical trials. And if a drug needs to be developed, certainly there's a lot of money that's going to have to be invested by pharmaceutical companies in that process. There's an in-between phase of between identifying a therapeutic target and being able to commence a clinical trial, which oftentimes in both in academia and industry is very difficult to get done. It can be very inefficient. It can be slow. Sometimes no one has a real incentive to do that work. So there's a gap that needs to be bridged. And in more common cancers or diseases where there's a lot more money at play and larger patient populations, either the market incentives drive companies to fill that gap, or there's a sufficient amount of funding, personnel, interest, et cetera, within academic institutions where, you know, that gap will get filled. In the case of an ultra rare cancer like chordoma, where there isn't a large financial incentive and there isn't a huge field, that gap oftentimes doesn't get filled. And so really important projects or concepts oftentimes stall out. And no one has a real incentive or sufficient incentive to address them. And so that's one gap that our lab is intended to fill. Another thing that the lab is intended to do is to make it possible for many more researchers and companies to bring their unique expertise, skills, resources, et cetera, to bear on this disease. So if you're, you know, a researcher, let's say, who has niche expertise in some particular technology or some particular approach, signaling pathway, gene, whatever, you're not an expert in chordoma, but we want you to apply your expertise to this disease. For you to start a chordoma program in your lab, there's a huge amount of expertise and knowledge you have to generate. You have to additionally generate capabilities in your lab and probably hire people and train them up. And so there's a huge impediment, a huge barrier that has to be overcome to be able to take on this disease, to apply your approaches and expertise to chordoma. We'd like to vastly diminish that hurdle, and the way that the lab does that is by serving as a kind of for bringing expertise in chordoma, so understanding about disease biology, but more importantly, developing deep expertise in a whole suite of capabilities that are chordoma specific, that can complement the kind of technology or drug or biology specific expertise that we want to bring to bear on the disease. So we can form these like very synergistic and constructive partnerships where we focus, we do what we do best, which is the kind of chordoma, the experiments in chordoma cells and mouse models, et cetera, and then partner with researchers who have like world-leading expertise in what they do best. And it's a very kind of complementary relationship. And then the final thing that I'll mention is that I guess in a similar vein with respect to brachyury drugs in particular. So I think many people have heard of this target brachyury. It is a diagnostic hallmark of chordoma, but it also is chordoma's, as far as we can tell, its kind of most significant vulnerability. There are no drugs currently that target brachyury, but I think there's wide recognition that this represents a huge opportunity for chordoma, also potentially for other tumor types as well. And so we have a major strategic priority for us is to encourage companies that have relevant technologies and approaches to undertake brachyury drug discovery efforts. The problem is none of these companies are expert in brachyury, and for them to set up those capabilities is cost prohibitive, or just there's infeasible or impractical in various ways. And so in order for us to make it a viable business decision for companies that even at the earliest stages of drug discovery to even take a look at brachyury, to take the very earliest steps in trying to develop brachyury drugs, we have to bring to them the capabilities to make it very easy for them to test those drugs in a biological context. And one thing I'll just mention is that people may have heard that in the last year, artificial intelligence has very much been in the news. But there's been a wave of artificial intelligence based drug discovery that has been kind of building over the last 5, 6, 7 years. And now there's like hundreds of companies that are employing AI or computational based drug discovery approaches to design or identify compounds that can interact with and serve as the basis for drugs against previously undruggable targets like brachyury. So all of a sudden in the last couple of years, the cost and difficulty of identifying potential brachyury drugs has just plummeted. However, all of those compounds that are identified or designed, they still need to be tested in the real world. They have to be tested in cells and biological systems, and the cost of that has not changed. And so basically, what we're trying to do is bring down the cost to companies to be able to kind of do all the preclinical work that's necessary to advance brachyury drugs. And ultimately, at the end of the day, companies need to make a business decision, and the business decision is based upon the size of the expected market, the time that it's going to take to develop drugs for that market, the risk and cost associated with it. And so there's different ways that we can change that calculus. We can help to demonstrate that there is a market beyond chordoma. That's one of the things we're doing in the lab right now. We don't know the answer, but we think that there is reason to believe that there might be a market beyond chordoma. And if we can generate stronger data to that effect, that changes the kind of economic calculus. But additionally, and importantly, if we can reduce the risk and the time and the cost of preclinical development, that totally changes the calculus and perhaps makes a business decision on behalf of a company that might have historically been infeasible now feasible. So I think we're starting to see evidence of that in terms of interest from various companies in this target.
Great. Thank you, Josh. We have a question came in through the chat from Renee. Renee, thanks for your question. Thanks for being here today. Renee is asking if lung metastases are discovered, do they tend to respond well to radiation? Or I guess maybe to make that question a little bit more broad, if a patient finds himself in this situation, what are some steps they should be considering?
Yes, that's a good question. And there isn't an exact answer, or there isn't one answer, I suppose I should say, that there aren't guidelines to say that lung metastasis should be treated exactly this specific way. The available treatments would be surgery, radiation, or drug therapy, and a lot of factors go into that. So there are a lot of ways that tumors in the lungs show up. Sometimes it might just be one tumor. Other times someone goes in for a scan and there are 20 small little 2 millimeter tumors. And questions have come up about this at our conferences during our Ask the Experts panels, almost at every conference, just what do we do with these? And consistently the doctors say that they point out that, of course, you don't know it's chordoma unless you do a biopsy. A biopsy could be risky, though. So unless the tumors are changing rapidly, growing rapidly, or are causing symptoms, a lot of chordoma experts would advise to watch them for a little while and not to rush to treatment right away or rush to a biopsy. Because if they're not going to change, then there isn't necessarily a need to do something about them. And what has been observed about lung tumors is that sometimes they pop up even in that situation of a number of them show up all at once, but then they don't change very much, and they're really not causing problems. And so there's no need to put yourself through a treatment if nothing's really happening. It would kind of be considered low risk, or there's not a lot of danger there. And then in some cases, if there are a number of small tumors, one might start to grow and cause problems. And so that one can be addressed by possibly surgery to remove it, or by radiation. If a lot of them are changing, if there are a lot and a number are changing and growing, if there are tumors in other parts of the body that are growing, then that's often when the drug therapies and systemic therapies would be looked at, because you have a number of tumor sites, and so it's difficult to address all of those through surgery or radiation. So a systemic treatment would be used to try to address all of the tumors, if possible. And even with drug therapies, surgery and radiation can still be used if you're being treated off-label, so not through a clinical trial. Surgery and radiation can be used if necessary. So I know that's not a very straightforward answer of this is the thing to do, but it really is dependent on the person's situation and what's happening with them, and then really finding the right way to address it. But the thing that always strikes me with lung tumors especially is the fact that they can appear and not change. And so that that can be a nerve-wracking thing to live with as the patient and the person who is dealing with this. But it's also, you don't have to put yourself through any treatments that are unnecessary if things aren't changing. So Josh mentioned multidisciplinary teams for new diagnosis earlier. And I think this is the type of case where a multidisciplinary team is just as important. So you're not newly diagnosed, but you should rely on the team of surgeon, radiation oncologist, medical oncologist to all assess the situation and work together to find out what path to go down, whether it's one type of treatment or a combination. So it's really relying on that team that has experience with metastatic chordoma to lay out your options for you and decide what's best for you.
That's great. Thank you, Shannon. Josh, did you have anything to add on that one?
Oh, it's perfect.
Okay, thanks. Um, Really good question in the chat from Royce. What does the latest research tell us about the ability of chordoma to be inherited?
Yeah, it's one that comes up a lot. I think the very short answer is that the risk of chordoma being passed on to a family member is exceedingly low. There have been roughly 10 families in the entire world that have been identified that have familial chordoma, meaning multiple members of the family affected by chordoma. Of those 10, a subset of those um, there's been an identification of the cause of familial chordoma, and it is that members affected by chordoma in those families have an extra copy of the brachyury gene. So whereas everyone ordinarily has 2 copies of every gene, these individuals have somehow a 3rd copy of brachyury. Again, exceedingly rare, and so I think the likelihood of for any one of us who are affected by this disease of having a family member with the disease, or a children or grandchildren being at risk is, it is vanishingly small. There is a related kind of genetic element of chordoma also connected to brachyury, and so this can be a little bit confusing, but I'll try to kind of explain it clearly. So whereas individuals who have familial chordoma have an extra copy of the brachyury gene, basically everyone who has chordoma has a different type of change in the brachyury gene. It is basically a single letter misspelling in that gene. And the interesting thing is that is not necessarily unique to chordoma. It's a relatively common variation or misspelling, what you call a variant in the gene, that is relatively widespread or pervasive within the population. I think something like 30 or 40% of people, and it varies depending upon background and location, et cetera. But roughly 30%, 40% of the population has this alteration in brachyury. In chordoma patients, it basically approaches 100%. So that tells us that there is some predisposition that this change in brachyury causes. But if you just look at individuals who have this alteration in brachyury, still the incidence is like, rather than being 1 in a million, it's maybe 2 in a million or something like that. So it's still very, very rare, even among that population. So there's no way to use this as a screening test, for example, for chordoma. It's just interesting and maybe tells us something about the biology of the disease, that yes, there's some role that brachyury is playing in the initiation of brachyury. And this is an area of very active inquiry. In fact, we just released a request for proposals last week to try to better understand basically how brachyury works in chordoma. So there's so much evidence pointing to brachyury as being the key driver and being a vulnerability, but why, what's it doing? Why is that the case? What is it interacting with? What are the changes in brachyury actually doing? These are really important questions, and not just esoteric academic questions, but they're really important questions because they could potentially point to new treatment avenues. So I hope that we'll know more about that, in the coming years. Oh, and one other thing that I'll just mention is that there's a couple other tumor predisposition syndromes that have been associated with chordoma. Again, this is very, very rare, but there's a tumor predisposition syndrome called tuberous sclerosis complex. Sometimes it's abbreviated TSC, and individuals with TSC are at an increased risk for getting chordoma. But I think in most cases, TSC would be diagnosed for other reasons. So you'd probably be aware if you had that, although there are some, I think, rare instances where there are kind of undiagnosed cases of TSC out there in the population. And there are, if there is any question whatsoever about TSC or other tumor predisposition syndromes, there are doctors who specialize in those. So there's even in certain places like a TSC clinic or kind of familial, tumor specialists, and if this question are concerned, they would be resources that you could consult with.
Awesome. Thank you. The chat is now filling up with questions, which I love to see. So thank you everybody for those. I'm going to get to as many of them as I can. Lennon, I'm going to start with you. And first of all, I'm really sorry for what your family is going through. So Lennon's daughter, a 12-year-old Catherine, is suffering from poorly differentiated chordoma. And Lennon, I know that your question is pretty specific about the path that you should take next. And so I want to encourage you, after Josh and Shannon maybe speak to this generally, for you to reach out to our patient navigators to get some more specific guidance, and they can learn more about what's going on with your family. But meanwhile, Josh and Shannon, the question is about what are the best treatments available for poorly differentiated chordoma? Chemo, radiation, is there a particular drug that seems to work best in that population?
Josh, you want me to speak on that? Okay, so it's a really good question. And here in the US at the National Cancer Institute, there's a team that is, they're running a natural history study on chordoma, and kind of a sub-focus of that is poorly differentiated chordoma, and working on treatment guidelines for that. But while there are no specific guidelines right now, there is kind of an agreement that that chemo should be part of the first treatments, and it can vary depending on the specific situation, but poorly differentiated, it's different in some ways than conventional chordoma. So it seems to, we've observed is that it reacts better to traditional chemotherapies than conventional chordoma does. And conventional chemotherapies, meaning there's treatment regimens that have been used for types of sarcoma like Ewing sarcoma, that are often used for poorly differentiated. And so the chemotherapy is usually, if it's known at the time of diagnosis that it's a poorly differentiated tumor, and no surgery has been done, and no other treatments, then chemo is often started first, and then a surgery could be done possibly if the tumor has shrunk some, and a surgery is feasible. Radiation could be brought in at some point, but there is a chemotherapy regimen, and we've got that on our website, that there are I think it's 3 usually that are combined. However, and Josh, you might remember this better than I do, but at the last chordoma clinic that was done at the NCI with the group I mentioned, where they invite patients with usually pediatrics, not necessarily poorly differentiated, but pediatric and young adult patients to this clinic that they host once a year. There was some evidence presented about the chemotherapies, and specifically a phosphamide, right, Josh? And the results that doctors have seen with a phosphamide in poorly differentiated chordoma. So I guess what I would say is, generally speaking, that's the path for poorly differentiated patients, that have, that it's known from the start that that's the subtype of chordoma that you have. But again, a team who can look at all aspects of the case and know when to bring in the different treatment modalities of surgery, radiation, and chemotherapy is really important. But it is, the subtype of poorly differentiated is one of the times where chemotherapy is considered a very viable treatment option and should be part of the plan in some form or another. So Josh, did you want to say anything else about the phosphamide or anything else?
No, I'll just mention that I think there is definitely anecdotes or evidence that can be highly effective, and lead to long-term disease control or stabilization, or maybe in some cases dramatic regressions. or even complete regressions. If the tumor progresses thereafter, it's possible that chemotherapy could once again be an option, but there's additional options to potentially be considered if and when a poorly differentiated progresses. So one potential option that has been evaluated in clinical trials, although still small numbers and the full results have not been published, but there's a drug called tazemetostat, where there's some evidence of activity and substantial clinical benefit in some cases for patients with poorly differentiated chordoma. And then there's also some anecdotal responses to various checkpoint inhibitors. So these are a class of immunotherapies that basically take the brakes off the immune system's response to cancer. And I believe that there are actually 2 ongoing clinical trials testing various checkpoint inhibitors in the context of poorly differentiated chordoma. One of them is being led by Dr. Mary Frances Whittakin-Malone, who Shannon mentioned was at the National Cancer Institute. And then one of them is being led by a group of investigators out of the Dana-Farber, I think it's Dana-Farber Cancer Institute, if I'm not mistaken. And I believe, Shannon, correct me if I'm wrong, but I believe both of those are on our website and could be worth discussing with your care team. But both of those trials, the eligibility requirements are basically dictated as for patients with advanced tumors, I believe, and wouldn't be relevant for a patient with a primary tumor.
And I think also, Josh, we should say that, Lennon, I'm not sure if you are in the US or outside the US and where you are, but tazemetostat is now approved for Ewing sarcoma.
Epithelial
sarcoma. So it is approved. It's FDA approved. I believe EMA approved. So it's, in that case, a drug like tazemetostat would possibly be available off label. So a medical oncologist could prescribe it. That depends on rules in the country where you live, and if you're covered by insurance rather than a national health system, whether your insurance will pay for it. But that is a way to get a drug like tazemetostat outside of a clinical trial if you are not able to participate in a clinical trial. And that's general information for drugs that are approved and not available through clinical trials. But, Josh and I are sort of talking about some things that are available in the US, and I'm not sure if that's where you're located.
So, in case if the tazemetostat can be given to a 12-year child, because I read that it has to be given about 16. So we are based out of India.
And that was about tazemetostat?
Yes.
That's a good question. I can look that up for you, and we can let you know. I didn't think that was the case, but it might be. And so sometimes there are restrictions for age. That's usually more for clinical trials, not the drug itself. But that's certainly something that we can look into and let you know about.
Okay.
All right, thank you. Next question is from John Gordon. So he says he's had a recurrence of his tumor 21 years after his first surgery. And I think his recurrence was 5 years ago. He's had a total of 72 radiation treatments to the area. And his question is, if it recurs in the same area, can you speak to whether he could be radiated a third time, or what about radiation if it reappears in the lungs or other parts of the spine?
I think the answer is it really depends. I mean, this is where there isn't like a one size fits all approach. It's so context dependent. It's so dependent upon the particular anatomy of your tumor, where it's located, where it is relative to the previous radiation field. So even if it's like adjacent, very close to, in the same vicinity as the original, where you originally had radiation, it's still possible that if it's on the edge of the radiation field, it's still possible that it might be possible to have additional radiation and not exceed tolerability limits for normal tissue. But that's really something that can only be determined by looking at the previous radiation that you've had, and comparing that to a radiation plan, or basically designing a radiation plan around that. So it really isn't a blanket answer. But whether you've had radiation if you've had radiation to one location, that shouldn't preclude you from having radiation to a distant location like lungs or elsewhere.
Okay, Shannon, anything to add on that one? Okay, thank you. The next question is from Sarah Powers. She asked, most chordomas are slow growing. Why are some chordomas rapidly growing? Are those considered to be more aggressive? Do they have a higher chance of recurrence or metastasis?
Yeah, really good question. It's a very active area of research right now, and we're specifically supporting projects that are comparing tumors that, again, are primary and versus metastatic, but also trying to include large sample sizes so that we can kind of compare tumors that have grown more slowly versus tumors that have progressed very quickly. But as far as I'm aware, and Shannon, I don't know if you've heard anything else, but as far as I'm aware, there's really nothing known about what might underlie different growth rates of the chordoma tumors. There are, I would say, however, like certain biomarkers that have been identified that can help predict which tumors are more likely to progress more rapidly versus more slowly. There's one that's been used for many, many years. It's not super precise, but I think there's a relatively strong association between what's called the proliferative index, or oftentimes what will be called Ki-67. So it's basically the percentage of tumor cells that are dividing at any given time. There's a correlation between that and how fast the tumor is likely to progress or come back. But perhaps more sensitively, and also more recently, there's been some biomarkers that have been identified by a group of researchers at the University of Pittsburgh that look at kind of changes in certain portions of the chromosomes within chordoma tumors, and they've found that if the tumors lose certain of these chromosomal sections, that is also associated with a greater propensity for the tumor to recur, as well as, interestingly, a greater propensity for patients to benefit from radiation. So that is probably telling us something about what's causing those tumors to be more aggressive, if you will, but the mechanism is unknown still. So we don't know why that association exists. We don't know why the loss of those chromosomal segments leads to the behavior that's been observed of these tumors progressing or kind of behaving more aggressively.
I think also that what Josh, you were just talking about was related to conventional chordoma, and what, like we were just saying about the question about poorly differentiated, the poorly differentiated subtype and dedifferentiated subtype are known to grow very quickly and spread for whatever reasons, but those affect 5% or less of chordoma patients in general, and poorly differentiated affects children and young adults much more often than older adults. But yes, then within conventional chordomas, there are different rates of growth, and that's the thing that Josh was mentioning, that we're trying to understand why that is.
All right, thank you. Question from Jean Coy. Can you speak to what is the latest in immunotherapy for chordoma? He also mentions he's experiencing a recurrence, and he's currently taking a drug combination, forgive my pronunciation, Opdivo Yervoy, and experiencing really good results with that.
Yeah, that's great to hear.
I'm really glad to hear that, Jean.
Yeah. So earlier I mentioned the drug pembrolizumab. That's another drug in the same class. They're called checkpoint inhibitors. Oftentimes it's called, it'll be abbreviated, or kind of shorthand, people will call it Pembro. And then nivolumab will be, Opdivo is the brand name for nivolumab. And so oftentimes they're called Pembro and Nivo, and people have likened them to kind of Coke and Pepsi. They're very, very similar, although not exactly the same. This is like very clearly an active class of drugs in chordoma. The largest study to date was a study out of France that was published earlier this year. I think it was about 30 patients were treated with Pembro, and the response rate was 12%, if I'm remembering correctly. But additionally, so that's by response rate, I mean patients whose tumors have shrunk by 30%. So Gene, it sounds like you're having a response, but it's would be called a sub-resist response. And that's still really meaningful, and it's like a clear sign that the drug is active. There are a subset of patients that stay on these class of drugs for a while. So sometimes the response can be quite durable, but it does vary. So I think there's more research that's needed on this class of drugs. Just like pemetrexed, this class of drugs is used in a lot of different combinations. So like that's the low-hanging fruit, is to combine those two in combination, but there's other combination opportunities with various checkpoint inhibitors. We've done some work preclinically to try to identify other checkpoint inhibitors or various combinations with checkpoint inhibitors, but it's very technically challenging to do those experiments, and very expensive to do those experiments preclinically. So probably to a greater extent than with targeted therapies, like things like afatinib or cetuximab, et cetera, to a greater extent with immunotherapies and checkpoint inhibitors in particular, there's a need to generate answers in the clinic, in clinical trials. And so because of the potential here, there is a kind of a whole slew of potential upcoming clinical trials, and a big need right now is to try to streamline the process of running clinical trials so that more patients can benefit more quickly from those concepts that we hope to bring into clinical trials, and we can get answers more quickly. So that's a big focus right now. And then I guess the final thing I'll mention is, beyond checkpoint inhibitors, there's lots and lots of other immunotherapies that are being developed. I mean, like hundreds and hundreds of different immunotherapies, and any one of which could represent an opportunity for chordoma. There's no way all of them are going to be able to be tried against chordoma. So we have to try to understand the biology. So not just the intrinsic chordoma biology, but the biology of the chordoma immune interaction. So like, what is chordoma doing to the immune system? What is the immune system doing to chordoma? How is chordoma rebuffing or kind of evading a destruction by the immune system? And we have a long-running partnership now. It's going on for 5 years with, it's called the Cancer Research Institute. They're the leading funder in the world, the leading private funder in the world of cancer immunotherapy research. They've been around for about 70 years. Funded a number of Nobel Prize-winning immunotherapy researchers. And so we partner with them to try to attract some of the world's leading immunotherapy researchers to chordoma to find new avenues of immunotherapy for this disease. So we've funded for 4 years now, 4 years running. We've co-funded projects with CRI. We've just announced a request for proposals for the 5th year. And this is a kind of a very active area of focus, and where we're really seeking to invest to the extent resources permit a lot more going forward.
So thanks. There's a question from Richard in the chat that I'm going to try to summarize, but Richard, if I misunderstand this, please jump in and let me know. To follow up on what you were saying, Josh, about brachyury relevant companies reaching out to the Chordoma Foundation saying, we have an idea or a drug that we want to test with brachyury. How does that work? When, do they reach out, and how do we make a decision about whether to work with them? And then the next question he had, which is, how many of those types of studies or partnerships can we take on in a given year?
That's a good question. It's a mix of our proactive outreach to companies, and then more recently, several companies coming to us. We are really only limited by capacity in the lab. And then, by extension, limited by financial resources to, enable that capacity in the lab. But typically, and this is this playbook is being generated right now because, like these relationships are, are kind of just coming to fruition in recent months, and there's several that are in the pipeline. But the way that we envision it working is, we want to enable anyone, whether it be a company, an academic investigator, whomever, to bring their expertise, capabilities, ideas to bear on this target. And so to do that, we're offering basically in kind, at no cost, the ability for investigators and companies to test a limited number of compounds that they've identified in an initial set of experiments. And the idea there is to see if there is enough promise to warrant further investment on the part of the company or academic investigator, and further investment, time, effort, et cetera, on our part. So basically to reach a kind of an early go, no-go decision as quickly as possible. We want to eliminate all barriers to that. So we're just doing that work on an in-kind basis as quickly and easily as possible. If the results are promising, at that point, both we and the company or the academic investigator have a decision to make as to whether we want to move forward together, which would involve more substantial investments of time, resources, et cetera. And the specific nature of, who's providing what resources and on what basis is going to be very context dependent. But the expectation is that there would be, contributions from both sides. And, certainly in the case of companies where there is a, potential financial reward for successful development of these drugs, like we expect, if we are enabling that the foundation is, that some of those financial benefits would flow back to the foundation in an appropriate manner to enable the continuation of our research efforts. But the specific details of that are very much kind of context dependent.
All right, great. Richard, did that answer it for you?
Yes. So it's a two-way street, basically, where you're dealing with the company, they're helping you, and you're helping them, basically. I would think.
Right.
Yeah. Right, thank you.
Got it.
And
Thanks, Richard.
more than anything, our North Star is, how do we get effective therapies to patients as quickly as possible, with underscoring the word quickly. So we have a real emphasis on speed. And so that's not, oftentimes our need for speed and our sense of urgency coincides or overlaps with the incentives and needs and interests of academic researchers and companies, but not always. Sometimes there are various kind of career incentives or economic incentives or other incentives that might drive the motivation of the priority to not be about speed, in terms of work that's done in academia or industry. And so I think an important part of the lab is bringing the urgency that we all feel to these projects, and that, because we are providing such critical and valuable enabling capabilities and resources, et cetera, it gives us the ability to kind of imbue our sense of urgency in these projects in a way that otherwise is not possible, and frankly is not possible if we're just providing money. So by actually doing the work ourselves, we're able to bring a level of urgency to the work that otherwise is very difficult, if not impossible.
Yep, great. Okay, there's one last question, and then I think we'll probably wrap it up there. For anybody who did not get their question answered or who still has more to ask, I'll post the email address in the chat after this so that you can follow up via email, and we're happy to help. The last question is from Sue, who wants to know, can you speak at all to the known efficacy of the use of cryoablation for chordoma-related lung nodules?
And you might have more direct experience with that. Any thoughts?
Well, there's very little official published evidence on cryoablation, and you probably know that, Sue. I think we have known a handful of people who've had cryoablation for lung tumors, and for the most part, what we've heard is, at least, I guess not for the most part, but what we have heard is that, at least initially, the patients have been very happy with the results. They have either had some, a bit of tumor shrinkage, or maybe tumor has died a little bit, but that's given symptom improvement. And that's important to the patients that have had those treatments. And so they were happy with it. I don't think we know questions like how long, if cryoablation seems successful at first, how long does that last before a tumor starts to grow again? I think all of that is still being looked at. But it's one of those options to consider, along with the other ones that we talked about earlier for lung tumors. And it's not just for lungs, cryoablation or thermal ablation, using heat or cold. Those are looked at for tumors on the spine as well, and can be options. And there are a few centers that are really looking at that in Chordoma more than others. And so hopefully we'll know more about it in the coming years, but it's certainly an option to keep in mind.
Thanks, Shannon. Thank you, Sue. Thank you, everybody, for your really good questions. Thank you for being here today. It was good to see you all, and we hope to do more of these in 2024. So stay tuned, and we hope you'll come back. Josh, or Shannon, any last thoughts before we let everybody go?
Yeah,
appreciate you guys being here.
Yeah, ditto that. Really enjoyed this. We're always available, so please don't hesitate to reach out anytime. And just so appreciate this community, your support for one another, support for the foundation. It really feels like we're all in this together. And thankfully there's a lot happening, a lot of progress being made. So I think a lot of reason for optimism. I certainly feel that. And I look forward to continuing to forge ahead with all of you. So I guess with that, we'll say goodbye, and I look forward to seeing you next time.